Hello, and thank you for joining us today. My name is David. I'm one of the doctors with ECS, one of the directors. I'm a consultant in emergency medicine and pre-hospital medicine. I'm sorry I can't be with you in person for a couple of reasons, obviously. The virtual world has become more apparent and appealing to us in current times, and also I'm at the other end of the country. So I currently work in the north of Scotland, in Aberdeen, where I'm coming live to you from. No, not live actually, recorded to you from. I hope that you're enjoying your day today. I hope it's of some use to you, and I thought we'd just give a little clinical update through a series of very short videos and updates. Um, Ed and I go through the notes and clinical interactions of ECS fairly regularly. And one of the most common interventions that we can deliver in terms of medicines is intravenous fluids. And sort of, I thought we'd have a little bit of a talk on fluids. I thought we'd base that on a bit of physiology, which is sort of the fundamental aspect of fluid delivery. Look at a little bit of the practicalities, a little bit of the law, and a little bit of clinical update. I'm not sure how much we'll get through. Um, Obviously, due to the nature of the technology we have, I'm not coming to you live. You cannot raise your hand. I will not see it. But if you do have questions, I'm sure Ed is available at during or the end to have a bit of a chat. Hopefully, you've had um, two handouts sent out. So the first one is a bit of just what we'll focus and we'll go through. So if you just make that available, and that's what I'll make reference to as we go through. And then number two, come to later on, probably one of the most useful things NICE have ever made, which is their IV fluid algorithms and when to give fluids, how to make an assessment of fluids. And we'll come to that at the end. Really a bit of a dry topic, but very useful. And we know that intravenous fluids, being something that the paramedic skill set brings to the mix, are really life-saving and really useful interventions if used appropriately. At times quite aggressive, at times quite cautious, but really with a bit of thought behind it rather than knee-jerk reactions of 250 mils of normal saline. So as we get started, let's think a little bit about the physiology. So if we take, for example, a 70 kilogram person, so I'm 70 plus VAT, but a standard 70 kilogram person is made up of around 65% body mass water. That means all that person, 70 kilograms, 65% of that is going to be water, a fluid, which is water. And there are variations within physiology within that. So as you get older, you dry out a little bit, so your percentage body water decreases. That's why you're more prone to dehydration. And when you're younger, you have an increased percentage of body fluid and less prone to subtle dehydration, and more that's when we've talked about when kids fall off the perch more in the past, and that plays into that a little bit. Those of us that are of a muscular disposition, we know that fat carries less body water than muscles, so muscular folks will have an increased body percentage water, and fat contains less. So those differences in physiology play into a little bit about where this water is found within the body. And that covers into body fluid compartments, and you'll see in your handout in front of you. So total body water, we talked about 65% of the body, and that will vary then depending on the weight of the person. 2/3 of this found in the intracellular fluid, so of all the cells that are made up, a bit of fluid will sit within the cells, and then everything that's not within cells is within the extracellular fluid, so everything that's outside of cells, extracellular. And that's then split to what's in the veins or in the vasculature, so veins, arteries, capillaries, and then what's not inside the vascular is the interstitium, so the in-between bit. So outside of cells, not in a vein or in a vasculature, is the interstitium. So intracellular fluid, you can see in the green on your handout, 2/3 of all the fluid in the body, and 1/3 is extracellular, of which that's split between vasculature, and the vascular we call plasma, and then the extracellular, extravascular, so that's the interstitium, what's in between. And you can see that's played out in the diagram in front of you, much easier to see than it is to explain. A couple of little facts about that anatomy that are just sort of key to hold on to. The split between intracellular fluid and extracellular fluid, so between the cells and outside of the cells, that split, that boundary is made up of a phospholipid bilayer. So the cell membrane is really almost impenetrable by most things, with a few exceptions that we'll come to talk about. But in general, anything that's large and anything that's charged can't get through there routinely. So what does pass through it routinely is water, can just move through it really quickly, but anything else either needs to go through a special gate, which we're not going to talk about too much, or stays where it is. So it's a really good boundary, the phospholipid bilayer, between the cells and outside the cells. The split between the vasculature and the interstitium, so the dotted line on your diagram, is different. So it's a semi-permeable membrane and allows things to pass through it very easily. So it allows water, but also allows ions or electrolytes that we'll come to talk about. So intracellular fluid, extracellular fluid. The extracellular is split up into what's in the vasculature and what's not in the vasculature, the interstitium, and the plasma. So what is there? If there isn't water, what else is carried? Well, obviously within the plasma there are red blood cells, there are white blood cells. We're going to put those to one side and not think about those at the minute. Inside the cells, you've got the nucleus and all the cell anatomy. We'll put that to one side and we'll think specifically, with regard to fluid, about electrolytes. So if you have a think about sodium chloride being that really common salt that we talk about, it's NaCl, so sodium chloride, and when you break that down, that becomes sodium and chloride. Both of those are charged particles, so sodium is a positively charged particle and chloride is a negatively charged particle. And charged particles on their own are called ions. When you put them together, they're called electrolytes. So sodium chloride is an electrolyte, is made up of ions of sodium and chloride. There are some ions that stay within the cells, and there are some that stay out with the cells. And I put that on your diagram or on your handout to make it really clear. There is no way of just knowing this. That is just the way things are. So intracellularly, you have potassium, you have magnesium, you have phosphate, and you've got proteins. Extracellularly, you have sodium, you have calcium, you have chloride, and you have bicarbonate. And those generally stay intracellularly and extracellularly. They can move between them through those special gates that we talked about. Put that to one side, we're not going to think about that. So generally, they stay where they are. So inside the cell, you have all the potassiums, the magnesiums, phosphates and proteins. Outside it, you have the other ones. And well, what's the significance of that? What's the relevance a little bit to us? Well, there's a little rule within it that these charged particles can't move. Remember I told you the phosphobilayer says what's large or charged can't move through the phospholipid bilayer. But you do then have concentrations and a concentration gradient inside and outside the cell. So in normality, in you or I, the concentration of all the potassiums, the magnesiums, the phosphates and the proteins are added up, and all the sodiums, the bicarbonates, the chlorides and the calciums are added up. And that number is called the osmolarity. So it's the concentration of the ions or the salts within a unit of volume, so within a litre. So all the potassiums, magnesiums, phosphates and proteins are added up. All the sodiums, the bicarbonates, the chlorides and the calciums are added up. And what happens is that actually that osmolarity adds up to be, in general, roughly 290. And that's just a number that just happens to be. If we have a think about that, so potassium, magnesium, phosphate and protein, or sodium, bicarbonate, chloride and calcium, when the 290 is equal inside the cell and outside the cell, there is no gradient, there is no concentration, and so water is freely movable between the two. Now what the cells don't like is when there's a concentration gradient, so when those two numbers aren't equal. And when things can't move by diffusion, they have to move by osmosis, and that's that process of water moving from an area of higher concentration to an area of lower concentration. and the phospholipid bilayer allows that to happen. Okay, hopefully you're still with us. That hopefully is pretty simple in that you have ions that are inside the cells, ions that are outside the cell, and water can follow the concentration gradient. So we've talked about in normality. On page 2 of your handout, you can see that you can see that in the middle of the diagram. So when does this change and what's the relevance to you? Because we're treating abnormality rather than normality. That's how the person's come across the radar. In normality, the person can drink and regulate their own renal physiology. So what they don't need, they excrete, and the kidneys are really perfect at managing these salts. But in abnormality, so in medical or trauma, burns or pediatrics, there's different changes that happen. So the disease process might be something like decay, it might be sepsis, it might be dehydration, and that's what we'll think about. And a couple of different patterns can happen, and it's really just thinking about what might be happening and how that might be playing out. So for instance, you can see my orange diagrams at the bottom of page 2. So in normality, in your extracellular water, in your extracellular fluid, you've got water and you've got the salts. We talked about which ones are extracellular. Mostly, let's think about sodium, because it causes most of our extracellular components, and it's the heavy-weighted ion. It's the ion that causes most of the weight within the calculations and holds the osmolarity. If someone is dehydrated, so let's for instance say that for 24 hours you didn't drink any water, and what would happen is that actually that concentration of sodium stays the same in the extracellular fluid. You're not taking in sodium, you're not losing sodium, it's all the same, you're just not adding water, which water at the tap contains no sodium, it won't add anything to it. And so the number of sodium initially will stay the same, but the amount of water and the volume in it will decrease. You've talked about this, about orange juice examples in the past, and I'm sure you can see the example on the screen is actually the amount of sodium stays the same, but the amount of water decreases, and therefore you become more concentrated extracellularly. So remember, outside of our cells, the amount of sodium stays the same, the water decreases, so the concentration, and remember we call that the osmolarity, that increases. So water follows a concentration gradient. So if we keep the sodium the same, but we decrease the amount of water, the concentration or the osmolarity will increase outside the cells. So water will try and move from the intracellular to the extracellular in order to balance it out. So water moves in order to balance the concentrations, and that's by that process of osmosis. So your cells, what happens to them is they then dry out and they shrink in size, and therefore you come to a series of problems. You lose lots of facilities that the cell does, and then the ions within the cells change, and you lead to a whole series of problems down the line that we'll come to. Let's take a little bit more of an example that's relevant to us with an ECS. Say, for instance, our event work, and we talk about excessive exercise and just how actually there's a little bit more complexity to this. And I've used the example of excessive exercise because actually it plays into two different outcomes that we find really difficult. So let's take that collapsed runner that we bring in. And remember, we've told you about sodium, lots and lots, and that actually it's really hard to clinically differentiate what the sodium is. So someone excessively exercises, let's say for instance they sweat off lots, and we know that sweat is really salty, so you're losing lots of sodiums, lots of chlorides through your sweat. And let's say you lose more sodium and chloride than you do the water that's following behind it. So then we've got that option 1, so loss in sodium in sweating in extracellular fluid, so the concentration drops. So extracellularly we lose all that sodium, the concentration of the sodium drops, and so that osmolarity, that's the concentration, the fancy name we give to that, it also drops. So water then moves into the cells in order to dilute the concentration within the cells. If the cells then get lots of water moving into it, you get lots of edema within the cells. And we know that if that in the skin cells, it probably doesn't matter so much, but the cells that are really important, say the cells in your brain, that can lead to cerebral edema. And that's when we get those patterns of low sodium because you've sweat it all out. Water then moves from extracellular to intracellular, you get cerebral edema, so they get reduced level of consciousness, they get that confusion and that agitation. And that's really what we're scared about. So we really fear hyponatremia due to excessive salt. what the body tries to do itself for that is then try to normalise it with your thirst and other protective mechanisms. So let's say option 2, excessive exercise, and you'd end up breathing a lot, so you become tachypneic, you lose lots of water that way, as an example, or you don't drink much water, and you try to keep your weight off, so you're not drinking much, and so the concentration of sodium then increases in the extracellular fluid, and so water then moves out of the cells. So sodium increases in the extracellular fluid, water moves from the cells to the extracellular fluid to try and normalise the concentration, and as a result the cells shrink and you get into that dehydration pattern that we talked about before. So within excessive exercise, we've got two options that could be happening, and that comes down to our use of the EPOC and the use of being able to build the ability to measure sodium in the context of exertional collapse, and that's why we put so much credence to it, because actually it could be high or it could be low. So giving you that little diagram there at the top of page 3, showing the intracellular, the extracellular, and the intravascular, we know that between the extracellular, the interstitium and the plasma, these salts can move freely, they can't move into the cells, so only water can move, and water will follow where the most concentrated salt is. So if there's lots of sodium extracellularly, but our salts all add up to a lower osmolarity inside the cell, water will follow that direction and lead to pathology that follows that by. Let's think, for example, a DKA type picture. Someone's glucose is really high within their vasculature. We know that the vasculature and the interstitium, they can just move between it, but the glucose can't go into the cells. You need insulin for that, and in DKA you have no insulin. So glucose is a really strong electrolyte, and water will follow. So if we think that the glucose is really high in the extracellular fluid, that's going to raise your osmolarity, and as a result water will move from inside the cells to the extravascular and into the plasma in this instance. So the person becomes dehydrated intracellularly, and they get all the fluid, all the water is following the glucose and going into the blood vessels, goes to the kidneys, and you end up peeing it out. And that's where you see that picture of polyuria and really dehydrated DKA patients. And so the treatment for them is, in the first instance, to get glucose, or to get the glucose, reduce the glucose and get it into the cells, start getting insulin using again, but also to rehydrate. The last example I've given you there is trauma. So let's think about blood loss. So we've lost blood, so we've lost the plasma. The extracellular plasma has just been lost with red cells. It's taken red cells, it's taken the platelets, it's taken everything, but it also takes the ions and salts with it. So the volume of water in the ions concentration remains the same, and so you lose, as well as your red cells, your sodiums, your chlorides, your bicarbonates, in equal volume. So the concentration doesn't change in the instance. It's like that juice, that cup of diluting juice, and you're not pouring the water out, you're not pouring the squash out, you're pouring everything out, and so the colour stays the same. So the treatment, therefore, isn't to give fluid, it's to give blood in that instance. So hopefully you followed that so far. I'm sorry that's a little bit of a little bit dry. That wasn't a pun. But you'll remember the intracellular ions are fixed there. We can't move them in and out very easily. The extracellular ions are fixed, but what moves is water, depending on where the concentration gradient. So all the intracellular ions added up, the extracellular ions added up, and then that becomes your osmolarity, and your osmolarity determines where water will flow. So where it is more concentrated, the body will try and dilute and balance those out to try and get an equal osmolarity, and that will move water by osmosis through that phospholipid bilayer from the cells either into the cells. So say in that instance that we talked about of drinking excess amounts of water without sodium in a race, and you might get that cerebral edema type picture, or in more commonly dehydration, DKA, sepsis type pictures. We'll pause that there and we'll move to the next video to look a little bit more at this.